Hello students, welcome to EPG Pathshala. I am Dr. Zeenat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to talk on the module Parenteral Control Drug Delivery Systems, and the focus would be the various infusion devices. This falls in the paper NDDS 1. The learning objectives of today's modules are summarized as: the first thing, we would be going in for the introduction, followed by what are the various properties of the implantable devices. Then we would focus on the performance criteria for intravenous infusion devices. Then we will spend a little time on highlighting the various advantages associated with them. We would also emphasize on the various types of infusion devices, which would include the osmotic pressure activated, the vapor pressure activated, and the battery powered activated drug delivery systems. Towards the end of the presentation, we would be in a position to talk about the recent innovations which are happening in the sterile drug delivery devices. And of course, the end would be the summary and conclusions. If we start to define the infusion devices, we take upon ourselves to define them as small body-mounted devices capable of delivering liquids such as drug solutions and other therapeutic agents for extended periods of time. It consists primarily of a portable infusion device in which the drug is mounted in a small separate pouch with pressure transmitting the driving medium. The drug is delivered from the system through the activation of an osmotic agent if it is an osmotic pump, and the rate of drug delivery is regulated by a rate controlling mechanism. Infusion devices can administer various pharmaceutical agents, which will include the hormones like insulin. They can very well be used for antibiotics. They are very well used for the chemotherapeutic agents, and also in situations where the pain relief drugs can be given, and most of the time when the patient is in a debilitated conditions, it is also be used for supplying of the nutritional supplement. Approximately, there are millions of infusion devices in use around the world every year, and probably the most of the usage happens in hospitalized or the hospital conditions. More than 80% of hospitalized patients receive drugs through IV route, which clearly indicates the important role of infusion pumps for providing preoperative care, critical care, and pain management. Infusion devices are also implantable devices which control the drug release at a zero order rate and allow the drug repository to be replenished from time to time. These systems consist primarily of a drug reservoir and a rate controlling unit, which in a layman's language or a common jargon can be referred to as a pump. It manages the volume of IV solution containing therapeutic agent administered to the patients. On the basis of the drug delivery or release monitoring mechanism, infusion devices are classified into the following types. We have the osmotic pressure activated drug delivery systems, vapor pressure activated drug delivery systems, battery powered drug delivery systems. Properties of implantable devices. The system capable of maintaining a zero order drug release rate by virtue of an inexhaustible energy source. This is a system which is biocompatible and allows easy adjustment post-implantation. The drug reservoir is small enough for easy implantation, but large enough to minimize the frequency of refill, which can be performed rapidly. Let us try to sum up the performance criteria for intravenous infusion devices. It is well understood that intravenous pathway provides an excellent route for the continuous drug therapy. The ideal delivery system regulates the drug concentration in the body to achieve and maintain a desired result. When the drug's effect cannot be monitored directly, it is frequently assumed that a specific blood concentration or infusion rate will achieve the therapeutic objective. Although under-infusion may not provide sufficient therapy, over-infusion can produce even more serious toxic side effects. What are osmotic pressure-activated drug delivery systems? Osmotic pumps are rate-controlled systems which provide continuous delivery of drugs stored in liquid or in the solid form. Due to the wide availability of water in all the body fluids, highly miniaturized implantable devices for even inaccessible body parts can be developed. Also, an efficient drug storage enables implantable device to provide constant drug release over a prolonged duration. In an osmotic device, the therapeutic agent in solution form is mounted in the semi-permeable membrane. The drug is released in a controlled manner at a constant rate by osmotic gradient. The rate of drug release is constant and is described by the following formula. dQ by dt is equal to 1 plus PWAM divided by HM into pi s minus pi e, where dQ by dt is understood to be as the rate of drug release, the PW is the water permeability, the AM is the effective surface area, the HM is the thickness of semi-permeable membrane, and the difference between pi s and pi e is equal to the osmotic pressure difference between the drug delivery system and the environment. My dear students, before moving on to the other systems in this category, we would try to sum up the various general advantages of the pump delivery system. The first and the foremost advantage which probably is there is that it ensures constant exposure to the test drug at a predictable level. It permits continuous administration of short half-life proteins and peptides. It provides a convenient method for the chronic dosing of laboratory animals per se. It minimizes unnecessary experimental variables and certify the reproducible, consistent results. It reduces handling and stress to laboratory animals. It allows a targeted delivery of drug or other agents to virtually any tissue. Because of being used continuously, it also has an advantage of being cost effective, and it does not require any electric energy. We now have a small discussion on the all-Z osmotic pump. All-Z osmotic pumps were developed in the 70s and are most commonly used today as mini osmotic pumps for delivering a large variety of drugs, hormones, and test agents with controlled rates for duration ranging from 1 day to 6 weeks without the need for external source. It is commercially available through Duoros Corporation, Cupertino, CA, USA. These can be implanted subcutaneously or intraperitoneally in various animal species. All-Z pumps may also be connected with a catheter to deliver the pump contents directly into the venous or the arterial systems, the brain, or into any other organs and tissues. All-Z osmotic pumps are cylindrical in shape, comprising of a collapsible reservoir composed of impermeable polyester bag with an osmotically active agent coated externally. The reservoir compartment is then sealed with a rigid semi-permeable polymer membrane of cellulose ester blend. A cannula serving as a flow moderator is inserted after filling the pump with the drug. The shape of the pump prevents the spilling of the pump's content and reduces its diffusive release. Also, the flow resistance obstructs the pump's content to be accidentally spilled out. This makes sure that the drug delivery is only controlled by osmosis. At the implantation site, the water content in the tissue fluid penetrates through the semi-permeable membrane to dissolve the osmotic agent, thus creating an osmotic pressure and resulting in drug release from the reservoir in controlled rate. The pumps are available in varying capacity of 100 microliters, 200 microliters and 2 ml and release rate ranging from 0.11 microliters per hour to 10 microliters per hour. Depending on the capacity and the delivering rate, the device can be made to work from 1 day to 6 weeks. Some technical points which are associated with osmotical driven pump can be summarized as: the osmotic pressure is used as an energy source. The drug reservoir is either a solution or a semi-solid formulation. It consists of a cellulosic outer membrane, whereas polyester intermembrane is also present. Students, in front of you is a typical structure of all-Z osmotic pump. You can see the different sections as just explained, which consists of the removable cap. It is then having an impermeable reservoir wall. Then you have the osmotic pump. The outer one is the semi-permeable membrane through which the water enters. Then you have the reservoir. The drug solution leaves through this reservoir after the pump is activated. All-Z pumps are most commonly used for prolonged drug delivery. It eliminates the need of repeated injections or tethered infusion systems or stress caused by these, which can result in ambiguous results and may affect the reproducibility of any clinical or preclinical study. Thus, it minimizes external stress and repetitive animal handling if it is used for preclinical studies, and eliminate the need for repeated nighttime or weekend dosing by lab personnel. It also delivers accurate dose, ensuring that all the recipients are properly dosed during the study period. It definitely allows the scientists to reduce the number of the subject or the animals needed to accomplish the statistical significance data. All-Z pumps can be used for in vitro studies where animals need to be replaced and controlled delivery is still required. It is also beneficial in case of human subjects, as there are no external connections necessary for infusions. It minimizes the risk of infection as compared to the externalized infusion systems. It also eliminates the possibility of a missed or a mistimed dose, overdosing and underdosing, and that is caused by repeated injections. We will now discuss the Duoros infusion implant. The Duoros technology was developed by All-Z Corporation, Mountain View, USA, which was later acquired by Johnson & Johnson in the year 2001. It consists of a non-biodegradable matchstick-sized cylindrical shaped device for delivering drugs for extended period of time ranging from 1 month to an year. An example is lioprolide acetate implant. The implant is inserted subcutaneously and retrieved at the end of the treatment duration. In this system, a protein or peptide is formulated into a stable solution or suspension, which is protected by the water-resistant, non-erodible, sterilized drug reservoir. The outer titanium alloy cylinder is capped by a semi-permeable polymer membrane at one end and by an exit port at the other end with an osmotic engine, piston, and the drug reservoir. When this system is implanted in the human body, water from surrounding tissue enters one end of the cylinder by the semi-permeable membrane, causing the osmotic engine to swell. This osmotic engine displaces a piston, which in turn causes the drug formulation to be released from a port at the other end of the system. The Duoros system pump conceptually resembles a miniature syringe in which the drug is pushed out in highly controlled form. Through osmosis, water from the body is slowly drawn through the semi-permeable membrane into the pump by salt, that is an osmotic agent residing in the engine compartment. The water drawn into the engine compartment expands the osmotic agent slowly and continuously displaces a piston to dispense small amounts of drug formulation from the drug reservoir through the orifice. The Duoros implant system can be used for systemic or site-specific administration of a drug. For systemic drug delivery, it is positioned just under the skin, that is in the upper arm, in an outpatient procedure that is completed in just a few minutes using a local anesthetic. Removal of the implant is also very simple and can be easily performed by a physician. Various therapeutic agents can be delivered through the Duoros implant technology, like the drug proteins, peptides, and genes, because the drug dispensing mechanism is independent from the drug substance used. Let us now dwell into understanding the characteristics of this just explained Duoros technology. Duoros can deliver drug up to 1000 milligrams of concentrated drugs from months to as long as an year. The engine can generate sufficient pressure to deliver highly concentrated viscous and non-water based formulations. The system can be engineered to accurately deliver a drug formulation as dosing rates down to 1/100 of a drop per day on a continuous basis and a continuous rate with a difference or standard deviation of plus minus 10%. The titanium shell of the system protects the drug formulation inside the body from the contact with the body fluids, so it protects itself from the enzyme degradation, water, and clearance processes within the body. So once it is being put inside the body, despite being in the tissue milieu, despite being in the biological fluid milieu, it still remains absolutely protected. The in vivo delivery rate is equal to the in vitro delivery rate, whereas the delivery rate can be controlled and maintained throughout the duration of usage. The first FDA-approved implant using the Duoros technology comes by the name Viadur, and the manufacturer were the Bayer AG from Germany. It was used for delivering the drug leuprolide acetate for a period of as long as 1 year for palliative treatment of advanced prostate cancer. We must appreciate it here that such systems are the ones which are most suitable for treatment in areas where a person suffers from cancer, which is probably requiring a continuous supply of the drug, and prostate cancer is one such condition. The behavior of the leuprolide acetate implant was extensively studied, and it revealed comparable in vitro, in vivo release rates. The device was very well accepted by patients. However, the manufacturing cost decreased the market demand of Viadur, which led to its discontinuance. The Duoros technology was also used in combination with sufentanil for treatment of patients with opioid-responsive chronic conditions. Another device developed by Intarkia Therapeutics Incorporation, Hayward, USA, for type 2 diabetes mellitus treatment with exenatide for up to 1 year is currently under phase 3 clinical development. Duoros pumps can also be used for drug release at target site by using a catheter which can be attached to the outlet port, guiding the drug release to the target organ or tissue. We will now shortly discuss the vapor pressure activated infusion devices. Vapor pressure activated infusion devices uses the vapor pressure as the power source. It is based on the principle that at a given temperature, a liquid in equilibrium with its vapor phase exerts a constant pressure that is independent of the enclosing volume. It basically consists of two chambers. Number one, an infusate chambers containing the drug solution. Number two, a chamber containing inexhaustible vaporizable fluid such as the fluorocarbons. These two chambers are well separated by means of a freely movable, flexible bellow. After implantation, the volatile liquid vaporizes at body temperature and generates a vapor pressure. The bellows move upward and forces the drug solution in the infusate chamber and release the drug through a series of flow regulator and delivery cannula in the systemic circulation at a constant flow rate. The equation related to the drug release goes as dq by dt equals to 3.1416 d4 delta p divided by 128 mu L, where dq by dt is equal to rate of drug release, small d is equal to the inner diameter, L is the length of delivery cannula, P is the pressure difference between the vapor pressure in the vapor chamber and the pressure at the implantation site, and mu is the viscosity of the drug solution. The vapor pressure infusate chambers have been successfully used to deliver the insulin for diabetics and morphine for terminally ill cancer patients. In front of your screens, you can see that it consists of the two chambers, which probably are the drug chamber and the charging fluid chamber. These are very well separated by the two set of bellows. After implantation, as we had just mentioned, the volatile liquid vaporizes at the body temperature and generates a vapor pressure. The bellows are then moving upwards and forces the drug solution in the infusate chamber and finally releases the drug through a series of flow regulator and the delivery cannula in the systemic circulation at a constant flow rate. You can also appreciate the presence of a bacterial filter which ensures that there is no presence of the contamination. The outer chamber is usually protected by silicone rubber coating, and primarily you can see that it is a very small cylindrical device which can be very well tolerated by any human system. The battery-powered drug delivery system is a second-generation programmed implantable drug delivery device. The infusion pump in this case is powered by a disposable or a rechargeable battery. The infusion pump has a housing having a recess. A motor which powers the pump is placed within the housing, and this is then connected to an electrical contact disposal in the recess. The recess is adapted to receive one of the disposable battery unit and a rechargeable battery unit. In case of the rechargeable battery, a transformer is used which is coupled to a conductive element for providing power from an AC power outlet. It also consists of a switch receiving the electronic signal to determine if the conductive element is providing power to the AC power source. The release of the drug molecules from the delivery system is programmed at a specific rate profile. This specific rate profile is actually predetermined and becomes a part of the design itself. This system controls molecular diffusion of the drug molecules in or across the barrier medium, within or surrounding the delivery system per se. Mainly, two types of battery-powered implantable programmable pumps are used, which are used successfully for delivering of insulin. These are number one, the peristaltic pump, and number two, the solenoid-driven reciprocating pump. Both are controlled electronically to release the drug at the desired site. They are designed in a manner that the drug moves only in forward direction towards the exit and never in the backward direction. In front of your screen is a very typical diagram of the battery powered drug delivery implant system. You can see the presence of the anode and the cathode and the movement of the positively charged drug molecules through the blood vessels. In this section of the module, we would try to focus on the recent innovations in the sterile drug delivery devices. The first one amongst them is the needle-free injection. Needle-free injections were developed by CrossChecked for subcutaneous, intramuscular, or intradermal routes. The device is suitable for all needle phobics and eliminates the risk of injury and needle contamination. Additionally, it improves the patient compliance and quality of life. We then have the Intervial Plus A. Duoros developed an advanced, simple to operate reconstitution device system which is designed for safety and precisely dissolve, suspend, and dispense a solid drug such as a protein or peptide compound. The system is an advanced form of the intervial system and differs from it in that it comprises of a lyophilized drug in cartridge and a diluent, usually sterile water for injection in a vial. On activation, the diluent from the vial moves the cartridge and dissolves the drug. The drug reconstitution is performed in its original container and delivered to the patient from the same container. Since the drug admixture is not transferred between the chamber, transfer losses are nil. The next type of the advanced systems are insulin pen injectors. Insulin pens are first introduced and marketed in 1985 by the name NovoPen by Novo Nordisk. They are used to inject insulin patients with diabetes and allows self-injection. Insulin pen comprises of an insulin cartridge either integrated or separate along with a dial for measuring the dose. These devices have a typical dosing range of 0.01 to 0.6 ml, that is 1 to 60 international units. These pens may either be pre-filled, which are thrown away when the insulin is used up, or have replaceable cartridges in 3 ml and 1.5 ml sizes, which makes it reusable. These can be used multiple times by changing the needle. However, it must not be used for more than one person. Insulin pens have several advantages over the conventional syringe, such as easy dose setting, late dose indication, patient compliance, ease of use, and dose accuracy. Regurgitation of blood may occur into the insulin cartridge after injection, which may lead to contamination and transmission if the pen is shared even when the needle is changed. The next one is the Monodose pens for dual chamber cartridges. Most of the parenterals are available as lyophilized formulation with dual chamber cartridges for reconstitution, and the device is disposed of after a single injection. The use of dual chamber cartridges puts special demands on the pen system in terms of spontaneous reconstitution, priming, and dose setting steps. It is very important for the patient that these steps are easy to learn and always performed in the correct order. The next one is the disposable auto injectors. These disposable auto injectors are usually single dose delivery devices designed for 1 ml long pre-filled syringes and are commonly used for infrequent injection of larger doses of drugs of different viscosities. The injection procedure, comprising of needle insertion, injection, and subsequent needle shielding, is performed automatically. Auto injectors developed by Somed's presents features like ease of use, full needle safety, and clear injection feedback. The next development is shown by something which is referred to as a pen with a memory. Humor Pen Memoir is the first and only insulin pen with a memory. Humor Pen Memoir records the date, time, and amount of the last 16 doses, including the priming doses, and helps to make a record of the amount of insulin taken. It allows setting of the dose by turning the dose knob in 1 unit increments up to a level of 60 units after the initial setup. Corrections can be made if too many units are dialed without wasting any insulin. It is a reusable pen for use only with Humalog, that is the insulin injection, which is having a recombinant DNA origin and comes in a 3 ml insulin cartridges. My dear students, we have finally reached to the last section of this module. Here we will try to summarize what we have learned in this particular module. We first got introduced ourselves to understanding what is an infusion device. We were able to appreciate that they are small portable systems which are highly suitable for drug delivery in situations where a continuous release of drug is required at a predetermined rate, and particularly in conditions for palliative therapy or for treatment of long-term chronic diseases like diabetes, mellitus, etc. We were able to specifically highlight the various properties of the different implantable devices, and we also summarized the various performance criteria for such infusion devices. Of course, we spent time in understanding the advantages, which are numerous for such systems. Then we spent a lot of time on understanding the various types of technologies which have been able to give us varieties of the infusion devices. We studied about the osmotic pressure activated devices, that how an osmotic agent is required to trigger the drug delivery. Then we probably went on understanding the vapor pressure activated drug delivery system here, where probably generation of a vapor pressure was emphasized upon for final drug delivery. And the last one we talked about was the battery powered activated drug delivery system. Towards the end of this module, we also tried to emphasize upon what were the various recent innovations in the sterile drug delivery devices. We had the needle-free device. We talked about the latest technology wherein we had a pen which was also supported by a memory, which simply implies that whatever time that drug delivery device is being used, it will not only deliver the drug at that point of time, and probably it will keep a record of how much of that was delivered at what dates and what times. This would probably come handy in giving the best possible care to a person who is suffering from chronic diseases like diabetes mellitus. In conclusion, we can say that these varieties of devices or systems are highly sophisticated, and primarily something which probably motivates us is that we are able to have the same situation where the drug is delivered continuously without having any amount of restrictions or barriers, and helping the patient to get the best therapeutic outcomes. Thank you.